Hello there, this is Lucina. I'm a PhD student from the Center for Neuroplasticity and Pain at Aalborg University in Denmark. And I will show you one of my PhD studies. As the title of the poster shows, my studies are focused on a non-invasive technique, which is Transcranial Direct Current Stimulation, or commonly referred by its acronym tDCS. So tDCS is a non-invasive neuromodulatory technique which delivers a direct current through the scalp over designated areas of interest in an attempt to modulate the activity and promote neuroplasticity of specific brain regions. There are a number of behavioral studies showing that tDCS of the primary motor cortex modulates pain thresholds and conditioned pain modulation, or CPM, in healthy pain-free individuals. But to date, there is no evidence of tDCS modulation during longer-lasting pain conditions. And in this regard, experimental pain models can play a major role in investigating and elucidating whether tDCS can promote advantageous plasticity, since experimental prolonged pain models comprise relevant clinical features such as ongoing pain, long-lasting hyperalgesia, altered corticomotor excitability, and reduced or impaired CPM. So, newly established paradigms have explored tDCS modulation based on the concept that targeting functionally associated areas can lead to a more widespread modulation than traditional tDCS protocols targeting solely one region. And also, to support this notion, neuronal networks respond to direct current fields even more sensibly than when targeting single neurons. One of these paradigms is the so-called multifocal tDCS of the resting state motor network, or in the short form, network tDCS, which has proved to increase corticomotor excitability more significantly in healthy individuals compared to traditional single-site protocols. Whereas traditional tDCS studies have demonstrated an augmented CPM in healthy subjects, the antinociceptive effects of negative tDCS have not yet been explored, in particular when assessing CPM impaired by prolonged experimental pain. So as you can see in the poster, in order to achieve the main aim of this study, 38 participants were recruited, 19 participants were allocated in each group, one in the active network tDCS group, and the remaining 19 participants were allocated in the sham group, which is the control condition. So as figure 1 shows, the network tDCS paradigm has 5 anodes. They are placed centrally on the left and right motor cortex. And we have also 3 cathodes. The cathodes are shown in gray. 2 of them are placed on the parietal cortex, and 1 of them is placed on the frontal cortex, specifically on FZ. So in order to produce prolonged burning pain for 24 hours, a topical capsaicin patch was placed on day 1 on the first metacarpus, and on day 2 it was placed on the center of the third metacarpus. Below the picture of the hand, you can find the experimental design of the experiment, which after the familiarization and the day 1 baseline assessment, the first patch was applied, and it was followed by a second assessment, which is day 1 post-cap. So this second assessment reflects the initial impact of capsaicin on the pain system. This assessment was followed by 2 sessions of tDCS. These 2 sessions were separated from each other by 24 hours. And after the second patch application on day 2, the final assessment was done, which is called actually day 2, and reflects the modulatory effects of 2 sessions of tDCS and also the impact of 24 hours of burning pain. So each assessment has the following outcomes. One of the outcomes is the pain intensity ratings, which were assessed using the numerical rating scale from 0 to 10. 0 is no pain and 10 is the worst pain imaginable. So pressure pain detection thresholds, or PDT, and pain tolerance thresholds, or PTT, were assessed on both the left and right legs for pain sensitivity measures using the calf pressure ergometry. And the CPM effect was also assessed through calf pressure ergometry, and it was expressed separately for the pain detection and the pain tolerance thresholds as a subtraction of the condition values minus values recorded for pain sensitivity at each time point. So all of these outcomes, like we were seeing in the experimental diagram, were assessed before the capsaicin application, so day 1 baseline, after the capsaicin application, which is day 1 post-cap assessment, and finally on day 2 after the second patch application. So let's move forward for the results. The most significant results can be seen on the poster. One of those is the pain intensity, which was exacerbated over time in both groups. As also the picture shows, there were no significant interactions, possibly suggesting that 2 sessions of network tDCS may not modulate pain perception. Another major finding was the CPM modulation during prolonged pain and due to the sessions of tDCS. There was a significant reduction of CPM effect considering the pain detection threshold between day 1 post-cap and day 1 baseline in both groups, suggesting that capsaicin-induced pain affected the descending pain inhibitory pathways. If we move to that picture 5A, but to the day 2, we can see that there were no significant effects when comparing to day 1 baseline and day 1 post-cap. This was most likely due to the different effects in the 2 groups. As we can see on that picture, there was a tendency for reduced CPM in the sham group and a tendency for normalized CPM in the active group. Furthermore, if we move to figure 5B, we can see the modulation of CPM considering the pain tolerance thresholds. And here there was a significant interaction in which post-hoc analysis showed a decrease of CPM effect on day 2 comparing to day 1 baseline only in the sham network tDCS group. The opposite happened actually on the active group, so there was a significant group difference only on day 2, suggesting that network tDCS can revert the consequences of prolonged pain. So as a conclusion, the results of this study suggest that network tDCS can revert or normalize the reduction of CPM elicited or provoked by prolonged pain for 24 hours, probably by engaging the resting state motor network. So this is the end of the presentation. Feel free to reach out, and if you have any comments or questions, you can find my email at the end of the poster. Have a nice day. Bye bye.